So I thought I'd talk to you a little bit about, sort of give you an overview of what I've looked at over the past sort of years, looking at somatic mutations in normal tissues, where we are at the moment, and maybe a little bit of a sneak peek into what we're planning to do in the near future. But before that, I thought I'd give you a little bit more of a sort of a backstory of how I got to be in the same Zoom room as you guys at the moment. And again, sorry I'm not there in person to do this. It's getting a bit hectic at the moment with moving and everything. But I started off doing an undergraduate degree in biology and physics back at the University of Utrecht in the Netherlands, where I'm originally from. And then, of course, as a young student, you're like, oh, I've got biology and physics up my sleeve. What the hell do I do with that? And so the natural step was to do a master's program in computational biology. And that's really when I touched genomics for the first time. And I worked with Professor Elizabeth Murchison over there on a very interesting and rare cancer in Tasmanian devils. Now, the weird thing about that cancer is that it's transmissible beyond its original host. So there's a clonal sort of transmission, if you will, of actual cells that go between individual devils, such that you have a cancer that actually clonally transmits between people and or between individuals and sort of goes beyond the original host, which is immensely interesting. But difficult to work with devil genomes at the time. And so I started doing my PhD then at the Wellcome Sanger Institute slightly later. And basically building on that fact that you can trace the ancestry of clones or lineages with mutations, I then started applying that to normal human cells at the time when this sort of normal tissue sequencing was very much in its early phases, but there was definitely a boom at the Sanger Institute at the time, and I completed that with Mike Stratton, Inigo Martin Carina, and Sam Bajati, and some of that work will feature today. I then thought, why not explore the other side of the Atlantic for a few years? And so I came to the Broad Institute, funded by an EMBO postdoctoral fellowship, and I worked with Gadi Getz and Kristen Artley basically up until now. Those My time here is focused sort of on contributing to two different consortia, which I'll mention at the end. One of them is somatic mosaicism across human tissues, or a very Bostonian SMART network. The other one is the Developmental Genotype Tissue Expression Project, DGTEX for short. And I'm only here for another few weeks or so, and then I'm spreading my wings and flying back to the UK, where I'll set up my group at the European Bioinformatics Institute, which is part of the European Molecular Biology Laboratory. Now, if after this sort of hour of me talking and answering questions, if you haven't been bored of me and you're looking for a postdoc somewhere, I am hiring, especially if you're keen to sort of escape the US for an undefined period of, say, 3 to 4 years or so. Now, to get back onto the sort of the topic central to today's story, we know that cells acquire mutations, and that's a process that starts as soon as the fertilized egg cell divides into its two daughter cells. Cells keep copying their DNA. With every once in a while, there may be an error, and of course, they can also be damaged through exposure to mutagens. Now, a lot of this somatic genomics that we know about came originally from sequencing the genomes of cancers. But as you can imagine, that's the endpoint of a long sort of life of mutation acquisition. And we knew relatively little about the early stages. For that, you really need to look at normal cells. And there's many questions that we can answer around that theme. How do different organs mutate? Is it at the same speed? Are there different mutational processes active in each of them? Can we learn something about development by looking at mutations that are shared between different cells, etc.? Now, of course, there's different types of mutations that I'll cover today. Those include the very simple single nucleotide variants, single base substitutions, whatever you want to call them, small insertions and deletions, all the way up to larger events like whole chromosome gains and losses, structural variation, etc. Although, truth be told, the data that I'm showing you today is mainly short read focused, and that will struggle sometimes with calling mobile element insertions or variants in more repetitive regions. More on that later. But like I alluded to, sequencing a cancer is basically seeing the result of a long evolutionary race. And the cancer, of course, is the winner in that regard. It's very, we think it's being, one of the causes of cancer is this stepwise accumulation of key driver mutations, key mutations that change the phenotype of the cell in such a way that it now becomes dysregulated and divides uncontrollably and gets the hallmarks of a cancer proper. But you may imagine that there's lots of other cells there, and they may be at various different points in that trajectory from normal cell to cancer cell. And so if we can take different snapshots of different people of different ages, can we find sort of a distribution of clones like that are already on their way to becoming cancer but aren't quite there yet? And what we've come to realize over the years is that maybe there's other pathways as well. Not every cell has to evolve into sort of this cancer-like trajectory. There may be other things at play, other things that select for different phenotypes of cells that may not result in carcinogenesis. But it's difficult to find these clones. If you think about just taking a bulk piece of tissue and putting it into a sequencer, you won't find very many somatic mutations, and that's because your resolution will drop dramatically very quickly. Imagine we doing 30x sequencing. Every heterozygous germline SNP, every inherited mutation basically, will be at a VAF of 0.5 in one of two chromosomes, and so in half of your reads. But if you start traversing this sort of linear, this sort of phylogenetic tree from the zygote onwards, which divides into its two daughter cells, its four granddaughter cells, etc., the variant allele frequency of these mutations that are acquired along the way, here, these colored stars, that will drop exponentially. The problem with that is that with the current error rates as they stand, you need to find a mutation in multiple reads to be sure that it's not just a sequencing artifact. And so very quickly, you'll hit your resolution, your detection limit over here, that you can't really go beyond variant allele frequency values of 0.07 in this particular example, or maybe a bit lower. Now, of course, you could just sequence deeper. Let's instead of 30x go to 300x, but you'll just delay the inevitable. At one point, quite quickly, you'll start hitting that exponential decrease again. But there are solutions, and we've come to realize a few of them. And so the two main groups that I'm talking about today is, rather than sequencing a population of cells, a polyclonal population of cells, if you will, we can sequence either single clones or single cells. The easiest or most direct way, I suppose, would be to imagine sequencing the DNA of single cells directly. Although there is not enough DNA to just get enough coverage to look at SNVs reliably. So what usually needs to happen is a form of biochemical amplification. And there's a few approaches out there that especially have gotten better over the past few years, including primary template-directed amplification. But I won't allude to that very much today. The other approach is to grow a single stem cell, usually, into a colony of cells. Let the cell do its own work of amplifying its DNA, isolate that single cell-derived clone, and then sequence that. Now, that's, of course, a bit laborious. You need to isolate these cells and grow them up in vitro. There could be some cultures there of mutations acquired, although we can usually filter those out quite reliably. It's just very labor-intensive, and it only works for a subset of cells. The work I'll present to you today is mostly focused around laser capture microdissection work. Now, that only works in a handful of tissues, but where it does work, it's quite beautiful. You can isolate individual tissue structures here, like a bit of this colonic epithelium. Here you see individual colonic crypts. You can isolate that one with a laser, and that population of cells will then be available to you to sequence. Now, I'll just, what's beautiful about that is that at that crypt, there is a single stem cell at the bottom that populates the entirety of that tissue structure, such that you're basically isolating an in vivo clone that's single cell derived, and you can then use your usual tricks of basically inferring that this genome was the genome of that single stem cell, and you can use it as a basis for your phylogenies, etc. This only works in select tissues, and usually these are glandular epithelial tissues where there is such a structure. Other examples are gastric glands, which I'll talk to you about today, and the endometrial glands, and a handful of other epithelial tissues. It gets complicated quite quickly if you go beyond tissue units, where you just sort of stab in the dark and there's maybe a sheet of epithelium like in the esophagus or skin. You sort of have to guess whether there is a clone or not. Luckily for us, a lot of the adult cancers come from these epithelial tissues. And so to answer some of the questions that we were interested in coming from a cancer genomics background, this approach was exceedingly well suited. A second sort of family of solutions around this problem is by lowering the error rate of sequencing. I'll come back to that at a later stage in the talk. And just to give you a flavor of the projects that then rely on this LCM data, this laser capture microdissection data, here are 3 different phylogenetic trees of these normal tissue clones that we isolated from 3 different people. And these span lots of different tissues. Again, mostly epithelial, but there's some exceptions to that. And there's a few things I want you to take away sort of almost immediately. If you look at this phylogeny over here, this is from an 80-year-old male individual. You'll find that the length of these branches, which is really the number of single nucleotide variants that are on that branch and hence and therefore in that sample, is quite variable between different tissues. All of these sort of dark gray dots, triangles, dots, etc., are glands of either the large or small intestine. Those have a very high mutation burden, a high mutation rate per year, if you will, while something like these blue dots, which are seminiferous tubules, which produce the sperm cells, have a very, very low mutation burden. And that's sort of in line with what we know from trio studies and other studies that we look at the paternal age effect that the mutation rate in sperm cells is incredibly low compared to sort of some other things that we know about. What this doesn't show you, of course, is what's happening at the beginning of the tree. This is basically a flat top with lots of lone branches, which makes sense. The last time something like a seminiferous tubule and a colonic crypt saw each other in terms of being the same cell would have been early in embryogenesis, and that's a very small part of this tree. But the information from that period is still in this phylogeny. If we forget about having the number of mutations as a branch length for a second and just go to unit branch length, if you will, they don't mean anything, you'll get a tree that looks like this. Again, this is for that 80-year-old male individual. We actually get quite an interesting and beautiful topology early on, where you can see that there's a single root of the tree, of course, and then it divides into 2 daughter cells, 4 granddaughter cells, and then it gets a bit messy later on. But really what's this showing is the earliest divisions of life. This is basically seeing the zygote divide into 2 cells and 4 cells, etc. And we can see those traces just from mutations. If you look for those mutations that are on that branch in different bulk samples from the same individual, you can estimate how much these these putative embryonic cells contributed to the adult body. What you'll find is that, at least in this individual, is that there's an uneven contribution. So this cell over here contributes about 2/3 of all adult cells in that body, and this one only 1/3. That's a pattern that's consistent across different tissues. Some exceptions are there, but then that asymmetry is not just there in the first cell division. You can also see it again here in the sort of second generation of cells, if you will, and that carries on. Why is that? That's a good question. We weren't the only ones to see this. There was a whole suite of papers around 2021 and before as well that saw this embryonic asymmetry from the first cell division across different human samples, many different human samples, also mice, where you see this consistent pattern of a daughter cell accounting for about 2/3 and another one about 1/3. Now, this is just a mean, or there's many exceptions and many different asymmetries, but it basically all comes down to the fact that we're not actually sampling every single cell that has arisen from the zygote. Very early on in embryogenesis, this is sort of a crash course in developmental biology if you've forgotten yours from a while ago. I never had a class in that, so I had to learn on the spot. But the zygote develops into this blob of cells, which then becomes the blastocyst. And the blastocyst has an inner cell mass over here, which will become the embryo purple later on, and forms this shell around it called the trophectoderm. The trophectoderm becomes part of the placenta. And of course, an 80-year-old man doesn't have his placenta around anymore, so we basically lose that part of the tree. But this is the first sort of cellular decision-making process that happens. And you can imagine that because we're dealing with low numbers of cells at that stage, 8 or 16 cells at that point, that if you just randomly start picking a number of cells to form the inner cell mass and another one to form the trophectoderm, that by chance you'll start getting asymmetries. This is almost like a game where you have an urn with black and white balls and you start picking from them. So basically what you'll end up seeing is this sort of probabilistic patterning of the early embryo, and you can figure that out with somatic mutations. We did some simulations in our data and some other people's data, and what we saw as the most likely solution to this problem is that at the 8-cell stage, 3 cells get picked to become the inner cell mass. Of course, 3 cells would always give you a 1/3, 2/3 asymmetry. But going back to sort of this earlier point about different tissues having different mutation rates, is something that we've come to appreciate sort of over the years from different studies, that you really have this scale from seminiferous tubules with exceedingly low mutation rate, the placenta, interestingly, with an exceedingly high mutation rate, and most normal tissues sort of sit in between. We haven't been able to quite measure this in oocytes just yet, so maybe that's a lower extreme than the sperm cells over here. But you can also find a few interesting things here. For example, if you look at neurons and hematopoietic stem cells, at least on the S&V side, they have a very similar somatic mutation rate, which is, of course, surprising. Neurons don't really divide, or don't divide very much, depending on who you believe. While hematopoietic stem cells, of course, more or less continuously divide. It depends on the quickness of the stem cells, but they have to replenish the circulating cells of the blood, of course, which at least in part seems to suggest that division rate doesn't dictate mutation rate. There's much more going on. Of course, I've only plotted the mutation rates here from things that look like endogenous mutational signatures, but more on that later. The other thing, of course, rather than just mutational patterns, is that we can also find signals of evolution, signals of somatic selection, really, for specific mutations that convey a phenotypic advantage. Most of these mutations that I mentioned before are in complete sort of regions of the genome where they wouldn't affect the functioning of the cell very much or are very quiet. But every once in a while, a key somatic mutation will hit a gene, and that will cause the cell to start behaving differently. We can actually use tricks from regular evolution, from bacterial evolution even, and this is something that in part was developed by Inigo Martin Corina at the Sanger Institute, one of my PIs, especially from his work 10 years ago on sun-exposed skin, but now a few years later in the esophagus, where you can start finding this selection for genes really that are associated with cancer. You can see TP53 here, Notch1, Notch2, etc. If you look at the esophageal epithelium of different people, now this is, you can imagine that this square is sort of a visual representation of that sheet of epithelium in the esophagus. You'll find little clones that have mutations in these key genes, and the size and abundance of those clones grows as we grow older. Now, there is a few things to note here. Those are mutations that if you look at them, you find them annotated, you would point at them in a cancer and say, this is what's causing the cancer. Yet we find them now in normal tissues where there's no obvious sort of pathological indication that there's something wrong with these cells. So they're very much not even pre-malignant, they're sort of still normal, but they start harboring these cancer-associated mutations. Other things beside aging can affect the abundance and size of these clones. So for example, here you see a heavy smoker in his 40s, which has a sort of a colonization pattern of these mutant clones that looks very much like someone much older in their 60s or 70s. So it's both, so the environment can definitely play a role, we think, in selecting for these specific clones. That pattern has been seen across different tissues, but that is interestingly different in different types of normal tissues, if you will. Skin and esophagus, I just mentioned them. They're both sheet-like epithelia, and they're actually quite similar in these selection landscapes. They both select for Notch1, Notch2, TP53 to some extent. There's a little interesting side story about Notch1 that I actually won't go into very much, but we actually find it more mutated in normal esophagus than in esophageal cancer. And that has led researchers down some rabbit hole, a quite interesting one, postulating that Notch1 mutant clones are actually, in a way, outcompeting pre-cancer clones, don't tend to form cancers very much themselves, and are therefore in some ways protective. We could talk more about that later. If you compare that to something different, like a more glandular epithelium, like the colon and endometrium, you'll find different rates and different drivers of different driver genes altogether. And one interesting difference, of course, here is the rate of how many cells or what percentage of your epithelium is colonized by driver clones at, say, age 60. This is a slide from Mike Stratton. Colon, for example, is exceedingly low. It's only 1% of glands that really will have a driver like that. While in the endometrium, at that same age, it's almost all of the glands will be colonized by something with a driver mutation. They're, of course, very different tissues in terms of their life cycle. The endometrium will go through monthly shedding and regrowth. And we think that has a huge part to play in selecting for these putative driver clones. But more on that later as well. One last thing before I'll go into a bit more depth into a specific project is that I wanted to show you that not only can these somatic clones underpin disease, as is the case with cancer, and we think with some other neurodevelopmental and neurological disorders, but there's also the other way around, where having a chronic disease or a chronic state of inflammation, etc., can actually influence the somatic mutation landscape. Here is a result from a paper from a few years ago from Peter Campbell's group, where they looked at non-alcoholic fatty liver disease, isolated these clones with LCM, with laser capture microdissection, and sequenced them and found something quite striking. A lot of these clones here, denoted by S22W or 21L, have mutations, the exact same mutations, in a gene called FOXO1. In another patient, that's also true, but for a different gene in GPAM. What those genes do is that they regulate lipid metabolism in the liver. Again, linking this to the non-alcohol fatty liver disease, what's basically happening here is that these clones get selected for because these cells have a preferential fitness advantage because they sort of cut down their lipid metabolism so much that they don't get exposed to the toxicity that's usually come with all this fat buildup in the liver. So all of that to say is that this chronic liver disease selects for clones with a certain phenotype that seem to be better able to survive under those disease circumstances. And you can imagine that this is sort of interesting in multiple ways, not just biologically, but also maybe clinically, if we can get some inspiration from these clones and how they deal with the disease, perhaps we can do better too. Now, the study I'll talk about for most of the time here is something that was just published a few weeks ago. It's the somatic mutation landscape of normal gastric epithelium. So I've talked to you about a few different epithelial tissues, just giving you a bit of a broad background about what we know about some of this. And now we'll zoom in on the stomach particularly. And I hope to convince you that there's some really exciting and really intriguing things happening in this particular tissue. Now, you probably all know what a stomach is, but it's the epithelial cells that we're interested in, and those are the glands that line the stomach. Those produce acid and enzymes that help basically make the stomach function, but it is also the origin of stomach cancer. And there's different risk factors to get stomach cancer: alcohol, tobacco smoke, obesity, etc. H. pylori infection is a big one, Epstein-Barr virus as well. An intriguing observation is that different parts of the world have different incidences of stomach cancer, and those correspond to differences regionally within the stomach where they come from. So, for example, in East Asia and South America, there's a very high incidence of stomach cancer, and that usually happens at the bottom of the stomach, the antrum, usually associated with chronic inflammation, while in Western countries where the incidence is lower, the rate of stomach cancer is highest in the region where it fuses with the esophagus, the gastroesophageal junction. So they have slightly different origins. But one important thing to note here is that there's this recognized precursor state or precursor risk conditions of chronic inflammation and intestinal metaplasia. Now, intestinal metaplasia is basically a fancy word of saying that a stomach gland starts looking like a crypt or a gland that you would see in the small or large intestine. So you basically get a level of tissue remodeling that seems to come prior to the emergence of stomach cancer. This study is based, again, on laser capture microdissection. We had at our disposal 29 donors in total, 17 with stomach cancer and 12 without. Those come from the UK, the US, and Hong Kong. And we asked those questions that I was alluding to earlier. What are the types of patterns of somatic mutations that we see? Do we see anything more esoteric with copy number changes or genes on the selection? And because of this cohort, do we see differences between cancer patients and non-cancer patients? Is there already something there in the normal cells that tells us that these people were at risk or anything like that? And this is just the right moment to acknowledge Grace, who did all the work with the LCM, and Mike and SY, who really spearheaded this project. Other thing to say is that this is the last project for my PhD. It's been about 3.5 years. So if there is a project from before that you haven't quite finished up, let this be an inspiration that there is a way out. And if you persevere enough, it will come out eventually. But this is the type of data that we get for these donors and these gastric glands. Here is a phylogenetic tree like the one I just showed you. And this is basically where we'll base all of our data analysis on. So here you can see different things like the number of mutations, how related these cells are, etc. If we look at the mutation burden, so just the sort of the length of these tips, and we just look at non-cancer donors, so those people without any gastric cancer there, we plot that mutation burden across age, you see a very beautiful and nice linear increase. As you grow older, you get more mutations, and that seems to be a linear increase. This is what we've seen in most other tissues. In stomach, it just happens to be the case that you get 28 SNVs a year and about 2 indels. Some outliers there, not always for a good reason, but we can talk more about those later. That puts sort of stomach firmly in the middle of this spectrum I alluded to before, so about the same as endometrial epithelium and a few others. Now, what's interesting is that these are the same plots I just showed you on the left. Those are from non-cancer donors, but if you start looking at those people with gastric cancer, you find something a little different. Again, this sort of gray box and this sort of dashed line is the same as just the average and the confidence interval plotted here for your convenience. And you can start seeing this whole cloud of samples of LCM laser capture microdissected glands that now have many more mutations than you'd expect for their age. And we don't see that really. I mean, there's maybe one hint of that here. Well, it could be for a different reason, more of that later. But we see these hypermutant glands basically just in these cancer patients. Why is that? And if we take a little bit of a closer look, we can use a few facts. We know exactly where we sampled these glands in the stomach. So here you see a little cartoon of the different regions of the stomach. And you find that basically all of these hypermutant glands are found in the antrum, the bottom of the stomach. The other thing, of course, is that we have H&E images of everything that we've microdissected. And so we can use pathologists and their expertise in annotating what's going on in that microenvironment of these glands that we've sampled. And so you can integrate pathology and genomics in actually quite a unique way and look at effects of chronic inflammation, and in our case, of course, also metaplasia. And what you find is that, again, this cloud here that's all in the antrum, they also experience relatively high degrees of chronic inflammation, but most importantly, many of them, if not almost all of them, are metaplastic. So they have already transformed from looking like a stomach gland into an intestinal crypt. And if you look at those glands with and without metaplasia, which is plotted over here, you can see that there's a very sharp increase in the number of SNVs that they have compared to their expected burden. So basically the distance to this dashed line, which is true for both single nucleotide variants and for short insertions and deletions. And of course, knowing where this comes from actually tells quite an interesting story. Here, I'm showing you a small phylogeny of just 6 samples, 4 of which have this intestinal metaplasia and 2 don't. And here you can see exactly where we've sampled those glands from in that tissue section. So for example, these 3 over here were sampled right next to each other, and you can see there is a relationship on the phylogeny. These have been sort of the same cell at some point in their life. But I think most intriguingly is this example over here, where we have 2 different stomach glands very close to each other. One of them has no intestinal metaplasia, and the other one has incomplete intestinal metaplasia. And despite being sampled sort of a quarter of a millimeter apart from each other, their mutation burden is in the fold changes of difference. This one with intestinal metaplasia has about 7,000 SNVs, this one without about 2,000. And again, a quarter of a millimeter apart, which just shows you how specific these effects are. So it's not just, we think it's not really chronic inflammation that's driving this increase in mutation rate or mutation burden. It's really the transformation into intestinal metaplasia that does it. And the other thing I didn't mention here is that it's not like that metaplasia is a single clone that's expanded. It's lots of different glands making that decision or being influenced into transforming into this different state. It's not that, it's sort of a pre-neoplasia or neoplasm in that sense. It's more of a field effect, if you will, although that term has different meanings for different people. I'll briefly mention mutational signatures here, but you may all very well be aware of that. Ludmil Alexandrov was, of course, one of the pioneers, and I think he's responsible for this color scheme, which I still like and use every day, basically. What this tells you is that you can look at distributions of mutations, in this case, single nucleotide variants, categorize them by their base change, so C to T or C to A, and the nucleotide that's flanking that change before and after, so within its trinucleotide context. Now, that distribution reflects some of the biochemistry that goes into creating that mutation, basically. And over the years, we've created this catalog of somatic mutational signatures, where, when possible, we've linked it to different causes. So smoking, for example, here is characterized by the C2A mutational signature. UV, which is sort of 7 over here, is very much C2T driven. And I won't go into the methodological part of that very much today, but suffice it to say that we've done the exercise of looking at this across many different normal tissues. One pattern that strikes us always is that most of the ones look incredibly boring, and they're all sort of a combination of these 2 or 3 mutational signatures. 5, which is sort of unknown and clock-like, but present in everything. It's sort of not quite clear what's causing it, but we've seen it in every cancer and normal tissue sequence so far. Signature 1 is this very particular spontaneous deamination of methylated cytosines. That's why you only see C to Ts and CpGs here. And another one that pops up every once in a while, especially in dividing tissues, is signature 18, which is linked to oxidative damage and the formation of adeoxoguanine, which then gets repaired as a C2A. We can combine the phylogenies that I talked to you before and then look at mutational signatures on this tree and see where the differences are. Is there anything different between metaplastic glands or not, or do they look the same? And you find these patterns. So, for example, this patient 1 here, an 85-year-old man, that signature profile looks incredibly boring. It's a combination of that 1, 5, and 18 I showed you on that previous slide. But once you start looking at gastric cancers, we've sampled a few here in this paper, you find a richness of esoteric mutational signatures that you couldn't really find in normal tissues very well. And the most intriguing one is signature 17. For those of you in the know, it's basically a signature that's only found in gastric cancer, esophageal cancer, and maybe a handful of other places. The exact cause is a bit unknown to us. There's some theories linking it to um, oxidation of the free nucleotide pool. Some people thought acid exposure for a while, but the precise cause is unclear. You see it in abundance in gastric cancer. I mean, you can sort of see how much that purple makes up this bar over here. It's many tens of thousands of mutations. We only find every once in a while a hint of that in normal cells. So here I've plotted this example of finding a little bit of that purple signature in some of these normal gastric glands. But it seems to be the case that for the vast majority of samples that we've looked at, these cells are protected from whatever mutations are underpinning signature 17. It may well be that as a tumor grows and that barrier between the cells and the lining of the stomach and the contents of the stomach grows weak or maybe even non-existent, that some interface there may cause an influx of mutations like that. But that's just me theorizing. The other thing to point out here is that in asterisks, I've put the glands with metaplasia, again, these hypermutant glands. And what you see here is that even by the naked eye, you can see that there's quite an increase in that blue signature, signature 18, oxidative damage, and signature 1, CTTs and CPGs. That remains statistically significant, fortunately. And it's actually quite extraordinary how much of an increase this is. So here you see the full increase in metaplastic glands compared to non-metaplastic glands. Signature 5, which is his background signature, stays roughly the same, and the increase is really driven by 1 and 18. The other place where we've seen this is in chronically inflamed IBD crypts from the large intestine, which are also sort of exposed to this chronic inflammation type thing. Another maybe wilder, but actually quite attractive explanation is that not only do these stomach glands start looking like intestinal crypts, their mutational landscape also switches. If you look at the mutational sort of distribution in colonic or intestinal crypts, you'll find also that it has more 1 and 18 compared to what you would normally see in the stomach. This doesn't just go hand in hand. This isn't just S and Vs on its own. It goes hand in hand with indels as well. And so in metaplasia and metaplastic glands, you see many more insertions and deletions. These are characterized by signatures known as ID1 and ID2, one base pair insertions and deletions at homopolymer runs. And it actually starts looking like the burden one might start expecting in tumors. You see actually an incredibly nice continuum between what's a normal gland, what's a metaplastic gland, and what's a tumor there. These are linked to polymerase errors. These ID1 and ID2 mutations slippage during the replication cycle. And so it may well be that these cells replicate faster, get more mutations that way. But the thing that I was most surprised by, besides this whole metaplasia and cancer precursor story, is the landscape of larger somatic variations in the normal stomach. And these are usually copy number changes, but I'll walk you through this. So we find a host of intrachromosomal events. These are large-scale deletions or large-scale insertions. Those will happen usually at fragile sites, so regions of the genome that, by nature of their sequence are predisposed to being deleted or duplicated. But what we also find are a whole host of trisomies. These are somatically acquired whole chromosome gains, and predominantly of a few key chromosomes, mainly chromosome 20, but 13, 7, and 18 also pop up multiple times. We find a few singular instances of copy-neutral loss of heterozygosity, but really by far the most unique aspect of this plot over here is this whole host of trisomies. And if you look at where they happen, that's this sort of bar down at the bottom, is that a lot of these trisomies are concentrated in a few individuals. Here are all the individuals of within the study, females over here, males over here, and they're ranked sort of ordered based on age. And you can see that even a few, it's not the oldest patients that always have these trisomies. There's a few of them here in the middle. They just happen to be, some of them happen to be cancer donors, but not all of them. So what's really going on here? The other thing I wanted to say is that, here, the burden of these trisomies is not significantly associated with age, which is what you would expect. And this is what we see indeed if you look at intrachromosomal deletions and insertions. And if you get older, and accounting for the number of samples we took per donor, of course, that you do get more and more of these mutations, which is normally what you'd expect with an age sort of related increase. But we can again use these phylogenies as a tool to look at these whole chromosome gains in a bit more detail. These are the two patients with the most amount of trisomies, and there's a few interesting things here. What I've plotted here in these red dots are events of that trisomy of chromosome 20. And what you can immediately see, because I plotted it like that, is that these trisomies happen multiple times independently in the same person. There's a few ways we can know that. For one, the actual copy of 20 that's gained is different. That's denoted by that 1 or that 2. But they also all had their own unique mutations that were required prior to that whole chromosome gain that got duplicated along with that. So they all have high VAF mutations in 2 out of 3 copies that are unique to each of these samples. And they serve as another confirmation that these are indeed different events. Now, the keen-eyed amongst you selves can also see this error bar around that. That's the timing estimate of when that gain is thought to happen. And you can get at that with the ratio of pre and post-duplication mutations. The earlier it happens, the fewer time it will have had to have mutations acquired prior to the duplication. So you'll see the vast majority being post-duplication mutations, while for lates, that's of course the other way around. And what you can see is that if you time each of these gains independently, you find that they happened around the same time. These confidence intervals all overlap, and in this case, we were able to bind it because of that sharedness between those two adjacent glands, which is interesting because, again, if we had assumed something was age associated, which these don't seem to be, you'd find some trisomies early in life, some middle in life, and some later in life, and you find this continuous process of accumulation. But this is not what we see at all. The other thing to note is that, which I'm not showing here, but I'll show you in more detail in the next slide, is that in some patients, especially the one over here, some of these glands, they do happen to have metaplasia, but they are confined to one biopsy of the stomach where we have taken multiple. So there is a bit of a spatial effect, which I'll show you in a bit more detail here because I briefly put it on the slide, but it didn't highlight it. Most of this study uses whole genome sequencing, but we did do a few targeted panel sequencing runs as well. And of course, knowing what the SNPs are of the chromosome 20 and how they're phased, we can see that because of some of these whole chromosome gains in some of these samples. We can then look at the panel data and ask with quite a lot of confidence whether we see a trisomy there as well. And we can use that then and then map that back to the epithelium where that sample came from. And so what you see here is that in sort of gold are trisomic gains of one haplotype, in blue the other, and it sort of gives you an idea of how dense this patchwork of mutant clones with just trisomies is in this donor. This is basically a centimeter across, this sort of bacon-looking like slab of gastric epithelium. And you can see that where we've sampled it, you see many different clones, even in close proximity to one another. Black is a clone where we didn't see any trisomy. So that means that quite a lot of this epithelium is colonized by these trisomic glands. What do? The last thing I'll say before that is that here we had indeed 3 different regions of the stomach sampled, and you can see it's not confined to just the fundus, the top of the stomach in this case, but it's highly enriched in it, especially when compared to the antrum and the body of the stomach itself. And there's sort of an interesting few things that I wanted you to take away from this, and I hope I managed to sort of highlight that from just the trisomy story, is that there doesn't seem to be a linear increase of these with age. They seem to happen around the same time in donors, and that's consistent across different donors. They can be sort of enriched or confined to single regions of the stomach. And this all screams at us that this is some sort of reaction to an exposure, some incident, an infection, if you will, that affected this person around that time in their life and left behind this genomic imprint of whole chromosome gains. We, of course, aren't around when that happens. We sample the stomach decades after this original insult happened. So we're not quite sure what's happening there. Of course, there's a few options here. Either this trisomy 20 is selected for, it gives these cells a selective advantage. That's one way, but it's difficult to pinpoint what exactly the phenotypic consequence is of that, because we're not confining it to a single gene, or it's not a focal amplification or anything like that. So I would say it's an unresolved mystery, but it's a very intriguing one at that. The last little bit of the stomach story is looking at point mutation drivers, a bit like what I showed you before with that bubble plot in the esophagus with the smoking story. What we find in the stomach is a set of genes that are under selection, positive selection, and those tend to be ones that are involved in the remodeling of the epigenome. So it's ARIT1A, KDM6A, ARIT1B, and ARIT2. But we find a few other interesting things as well. There's a hint of beta-catenin here. For those of you who know, that's usually what you need in a cancer for beta-catenin to sort of contribute to carcinogenesis and an activating mutation, quite often an in-frame deletion of S45, if I remember correctly. But that's not what we see here. So here in the normal stomach, the mutations in beta-catenin are inactivating ones, usually frame shift mutations or nonsense mutations at that. So that's a very different picture from what one would expect if you'd only sequenced cancers. And if you do the math, it's roughly between 5 and 10% of the gastric epithelium will have a driver by age 60. So it sort of fits more towards the colonic side than towards the endometrial side of this picture. We can, of course, map these clones, these driver clones in space, a bit like what I did before. And you can, for example, see that here there's quite a big clone with a double hit, one in KDM6A and one in ARIT1A. Here, what there may be a hint of is that there's two clones of beta-catenin mutations right next to each other. There's actually a third one just outside. That may highlight that there could be some form of a local selection pressure as well. Of course, thinking about selection pressures as sort of large-scale sort of exposures or influences like inflammation or maybe tobacco smoke, but there can be, we think, at least some hints of local exposures as well that change the selection landscape quite a bit. And that's an interesting one because in many of these gastric glands, if they're not metaplastic, they have a normal mutation burden as one would expect. And I didn't tell you this just about yet, but the glands that have a driver mutation, the glands that have metaplasia, and the glands that have trisomies don't really overlap more than you'd expect by chance. So these all feel like or seem like independent pathways these gastric cells take throughout their life in their journey of somatic evolution. And some of that, especially with the driver mutation, may also be that you get this dysfunction or this selection pressure, not through mutagenesis, but you get this driver landscape because of differences in selection. And there's other studies now out and about in the world being prepared that look at this in much greater detail. And they sort of find a whole host of carcinogens that don't act as mutagens, but may act as promoting agents or, if you will, compounds that select for a certain host of genes or mutations. That's where I was going with the stomach story. So I think I've mentioned all of this, but we find these hypermutant glands associated with inflammation, especially metaplasia. That in part can explain why they act as a precursor lesion. By upregulating their mutational rate, these glands maybe, they get, besides just being dysregulated, they may also be more prone to getting more mutations in the future. Their mutation rate's higher for SNVs, indels, but also structural variants. We find these glands with driver mutations in beta-catenin and epigenome remodeling agents, and of course these widespread trisomies, all slightly different journeys that these cells take. I sort of want to stop there talking about the stomach. I think I'm still on time. I'll quickly run through some of the future stuff in a few minutes, and then we have some time for questions, I think, unless there's already a burning question I can answer now. But what I wanted to talk to you about, at least in a little bit, is this network that I alluded to in the beginning called the Somatic Mosaicism Across Human Tissues Network, which just started in 2023. So we're already 2 years in, really. But it was set up to do this sort of research at a slightly larger scale with more people involved and more donors and tissues, etc. So the aim of the network is very much like what I talked to you about today. It's sort of discovering these somatic variants and figuring out what the patterns are in different kinds of healthy human tissues. Quite an exciting angle of the network as well is a whole host of projects devoted to tool and technology development. And these really are pushing the envelope, if you will, in improving our ability to detect these somatic variants either in small populations of cells or lowering the error rate so much that we can do duplex sequencing at an interesting scale, as I'll describe in a bit. And of course, then lastly, this data needs to be synthesized, put together for everyone to use as behooves an NIH consortium of this scale. It's a big network. It's across many different sites of the United States, with a bit of a concentration here in the Northeast. Five different sequencing centers are involved. It's about 300 people that are usually at these annual meetings, and it's quite a lot of fun, really, to work with all these different groups. I alluded to this. The main data set will be 150 donors between the ages of 18 and 85 or over, with lots of different tissue samples spanning the three different embryonic germ layers to look at more patterns of developmental mosaicism as well. I'm part at the moment, and going forward as well, I'll stay part of these two smart teams from the Broad Institute, one of them focusing us as a genome characterization center, a sequencing center really, and the other in tool and technology development space. The efforts of the GCC here are really devoted to scaling up duplex sequencing and making that work. So a few more words on what that is next slide, while we're doing some really interesting spatial work with the TTD group. I've alluded to this enough times to now finally it warrants its own slide. Duplex sequencing, for those of you who don't know, is a clever way of using the fact that a DNA molecule, of course, has two strands, double-stranded. And so if you were able to combine the information from both the forward and reverse strand of the same DNA duplex molecule, you'd effectively would be able to sort of lower your sequencing error rate quite a lot, because now a true mutation needs to be present in both of those strands, while any artifact that's generated throughout library preparation or sequencing, etc., would only be in one of the two. And that, ever since sort of NanoSeq, which is the Sanger flavor of this approach, UMI-based sort of duplex sequencing, came out in 2021, that's sort of blown the field open in some of these aspects. Now we can get down routinely to error rates down to sort of 1 in 100 million, 1 in a billion, maybe even lower than that. Depends a bit on how you measure it. That works very well for us because somatic SNV rates are in the order of more like 1 in a million to 1 in 10 million. And so your signal to noise ratio is much more favored on the noise side, on the signal side of the equation rather than the noise side now. What that means is that you can basically start trusting single molecule mutation calls, and therefore you don't really need, if all you wanted was an average sense of the mutational process, the pattern, the burden, etc., in your tissue, you don't need to go through this arduous process of either laser capture microdissecting or single clone expansion or even single cell DNA sequencing. But it works very well for short variants, SNVs and indels. Doesn't work very well for copy number changes or anything like that. And there's a whole host of different flavors of that basic principle that we're applying within the smart network. And I'll hope to update you at a future thing about how that's all going. But so far, the initial results look extremely promising. The second thing we're doing at the Broad here is what I said is sort of this more spatial aspect of exploring somatic mutation landscape. This is sort of taking the work with the laser capture microdissection and pushing it into the next level of resolution, maybe even looking at tissues where we couldn't look at so far because they don't have a clonal architecture like the one we need. This is an approach very much based on slide tags from Fei Chen's lab here at the Broad, where you basically overlay a tissue section with a barcoded array. Barcodes get cleaved, taken up by the nuclei, and then what you'll end up with is intact nuclei that are barcoded by their location in space, which you can then use for things like single-cell RNA sequencing, but in our case also for single-cell DNA sequencing. And what that then, this is not real data. This is just a plot showing what could happen with the data if we have it. But what you can imagine then is if you sequence lots of different cells, this is not even that, we shouldn't forget that whole genome sequencing is still expensive, so this isn't quite scalable if we want sort of deep genome coverage per cell. But you can start thinking about sampling these cells in space and then assigning clonal fields after you do phylogeny building, mutation filtering, etc. This is very much a focus of my life at the moment and also going forward at the EBI. Very last slide, I just want to give you a sense of scale of the somatic variation that we're dealing with, really. And my favorite example here is T cells. We've got about 10 billion of those, and we know that the rate's about 22 to 25 SNVs a year. If you do the back of the envelope calculation, that means that just your T cells alone acquire 8,000 SNVs a second. And that means that over your whole lifetime, every single SNV happens many multiple times, hundreds to thousands of times in just your T cells. Now, we're much more than just T cells, of course. We're many different cell types, and we have many different kinds of mutations beyond SNVs. So all of that to say that beyond just cataloging this somatic variation, what we really need to be doing is understanding the origins, their patterns, and their consequences in sort of mapping out both the trajectory from normal cell to cancer cell, but also any other role of somatic variation that we may have in our bodies, be that health, aging, or other diseases. With that, I think I would like to acknowledge all these people on the slide, mentors current and mentors past, everyone involved in the consortia, and of course you for listening. Thank you very much.
Thanks so much, Tim. It's really incredible work. There are several questions in the chat.
Oh, great.
The first question is from Joe Gleason. He asks,
Great.
there's a debate about what constitutes aging.
Yeah.
You mentioned several correlations between aging and mosaicism. Uh, is mosaicism the same as aging? Does mosaicism cause aging?
Yeah, we're getting right at the heart of the debate. That's great. So there's a few conflicting things out there, and I think a few nuances to this debate. So there was a study I was part of at the Sanger a few years ago, which looked at people with inherited polymerase deficiencies. Now, those people have a very high mutation rate, about eightfold higher, sixfold higher, although it's lower in the coding regions. They get more cancers. They don't, I mean, it's difficult to quantify aging per se, but they don't show any signs that we would associate with premature aging. So it may not always be that mutagenesis is the pathway through to aging, but what may well happen is that it's a bit more subtle. It's sort of, let's see if I can go back. Oh, this is back all the way though. But maybe it's a bit more like this, where sure, mutagenesis sets the stage for aging and all of this, but you need some sort of to go through these selection pressures to really start enriching your tissues for mutant clones that then create sort of larger scale dysfunction. Again, this is ongoing stuff, and sort of we don't quite know what's all the details here. But in my sort of limited understanding, I suppose, of what aging is, I think there is a role for somatic mutations, but maybe not in the most obvious way that you can imagine. I think it's a more complex interplay between both getting the mutation and then selecting for giving certain clones an advantage.
Thanks, Tim. So the next question is from Jung Ho Kong. He
Mm.
asks, do you envision using your findings for early cancer detection, possibly using circulating tumor DNA?
Yeah, it's a very interesting one, and it's I think the answer is yes and no. The interesting thing with circulating or cell-free DNA is, of course, that you get sort of a sample from the entire body in a way. You don't quite know where those molecules came from, although you can do good studies with looking at sort of the nucleosome imprints on cell-free DNA, so maybe you can have a good guess at it. I think what we've come to understand is that lots of these different normal tissues have mutations that we would associate with cancer. So almost rather than providing a way to do early detection, complicates pictures because now if you find, say, a TP53 mutation in your cell-free DNA, does it come from just a normal clone that's shed a bit into the blood, or is it more an actual tumor? And I don't have a good answer for that. So yes and no is the answer, whether we use this for cell-free DNA detection.
The next question says, can you please comment on mitochondrial DNA and somatic mutations in early cancer?
Yeah, I'll be brief about that because I don't really study mitochondrial mutations myself very much, but lots of people around me have. We went through the exercise of basically building these phylogenies if we only have mitochondrial mutations. That wasn't a great resolution. In terms of pathogenic mitochondrial mutations, we've much less looked at that. There may be a role for them. But that's part of what we're trying to do with SMART now as well. I didn't allude to that, but within that network, we're doing much more long read sequencing as well. And you may be able to get, entire mitochondrial haplotypes almost in one fell swoop with a long read. And so maybe there we're at a better resolution to look at some of these questions rather than with the short reads at the moment. Although there are people who look at these patterns and there is some interesting somatic evolution there.
Jung Ho also asks, can duplex sequencing work on genomic region with loss of heterozygosity?
Yeah, so it'll work on any part of the genome. That's not the problem. The problem is that it probably won't detect low-level copy neutral loss of heterozygosity or low-level losses. especially because mostly when we apply something like whole genome duplex sequencing, we don't go very deep. What we end up doing is going to 1 or 2x coverage for the entire genome. And if that's a bulk sample and any copy number change that you may have seen there would have been at a low fraction of that, it's difficult to pick that up from a signal like that, especially if you don't expect it to be there. If, for example, you had this sort of trisomy 20 story and you do low pass coverage, but you know that there's so you have phased haplotypes and you can figure it out, that's maybe better. But so far, it's the realm of duplex sequencing and structural variant and copy number calling hasn't quite been bridged.
Hector Chavez asks, which of the duplex methods do you think is best for in vitro cell culture for
Yep.
detecting somatics, SNVs, and indels?
Yes. So given that you asked for both SNVs and indels, I can tell you that two of them sort of drop down the list. There's the long read high def seq, which doesn't do very well with indels, and there's Ultima's PPM seq, which doesn't do very well with indels. The other four, those being NanoSeq, Codec, ComDuplex, and MetaCS or VistaSeq, all actually perform really similarly if we do it on any sort of bulk tissue or any sort of population of cells and look at their burdens and mutational signatures. So those are very yeah, very concordant. There may be slight differences in input requirements, input material, or lab expertise, but they all perform well. Is the spoiler there?
Oh, then this next question asks, something you actually didn't touch upon in your talk. Are there any regions of the genome that are more prone
Mm.
to getting higher mutation rates? Um,
Yes. Yeah, I didn't get into
Regulatory
that. But
regions.
yeah. It's but it's a very good point, and we do know this, especially from sequencing cancer genomes, and I think Ludmil must have done quite a lot of this in the Peacock days with looking at how different mutational signatures correlate with things like replication timing, transcription activity, transcription strand as well, of course. People here at Gaddy's lab have done that too. And you'll find that it's sort of slightly specific to different mutational signatures. Some really increase their prevalence with replication timing. Late replicating regions have less time to repair DNA damage, so there's more mutations there. Others are more prone to getting transcriptional damage, but maybe also transcription coupled repair. Um, and curiously enough, we've seen quite a lot of enriched um, signal in genes that are exceedingly highly expressed in some tissues, like albumin in liver, lipase in the stomach, surfactant in the lung. Um, this was first described, I think, by Matthew Meyerson and Marcin Emeliński back here at the Bird in 2016 or so. But basically, that's when the expression machinery, the transcriptional machinery, starts colliding with the replication machinery. And you get this sort of weird RNA DNA loops that then cause a lot of indels and SNVs, is what we think. So there are determinants of mutation rate locally. And I could devote another whole talk to that, I think.
Have you seen any correlation with gene length in your data?
Uh, Not so much, I think. Usually gene length is a problem because especially in something like DNDSCV, you need to count for gene length. You see more mutations the longer a gene is. And funnily enough, one usual red flag that something is quite wrong is if you see titin in your selection panel, if that pops up, because that's the largest gene, it's usually a sign that you didn't do something correctly. Um, so normally we see gene length as sort of a bad thing rather than a good thing. Other than that, I don't recall seeing something critical between gene length and somatic mutation rate. It's more the expression status that does it.
And then Ruben asks, at any point in beta-catenin, did you see any mutation drivers in addition to D32, S33, and S37?
Yeah, so funnily enough, the mutation that we see in beta-catenin tend to be frameshift indels and a handful of nonsense. So it isn't quite compatible with the cancer landscape that we would expect. Um, from memory, but it's in the supplement of the paper. I think a good fraction of those mutations were frameshift indels. Oh.
Great. And then I actually had a question kind of following up about Joe's point.
Yep.
Um, I think this is like a really important question in the fields. What is kind of like the causal, uh, I guess, path between aging, cancer, and somatic
Mm.
mutations. And um, I'm curious if you think, it seems like you're kind of measuring the state of the tissue using like these signatures as well as some of the pathology measures.
Yeah.
Um, do you think kind of collecting additional genomic information about the transcriptome and the epigenome, maybe with this new technology, the slide type technology might get at that?
Oh yeah, absolutely. I think the avenues that I'm most excited about are methylation, having that sort of almost in parallel to some of these studies. And there's definitely work. Well, Peter Campbell's no longer at the Sanger, but when his group was still there, they were devoting a lot of time at looking at methylation patterns and how that coincides with somatic evolution and what one can learn from that. And that may also be some of the hidden things that, maybe, the other way an exposure could work is by not inducing a mutational change, maybe not even selecting for things, but inducing an epigenomic change or something like that.
Mm.
We don't have any visibility on that at the moment. And then, of course, the other readout is asking the question, now, if I have a normal cell with a Notch1 mutation or TP53, is there already a difference in expression level in certain genes or in certain pathways that pushes them away from that normal state into maybe a precancerous one? What's the real phenotypic effect of some of these somatic mutations? And that's also difficult to answer. For that, you really need paired data of knowing which cells are mutants and which ones are normal and having their RNA. And I think work that Dan Landau is doing over in New York is sort of quite exciting there. I forget what the acronyms are, gotcha, and a few varieties of that, but really looking at the effect of mutations on the either RNA or epigenomic landscape. Yeah.
And then Michal asks, referring to the concurrent trisomies finding,
Mm.
um, I guess due to one trigger in time, has anyone investigated what might this event be?
No, we haven't really. Partially because we just have these samples, from donors and individuals, and basically do observational studies, if you will, to see what we find. What this needs, I mean, is some good environmental sort of studies, I think, and looking at maybe even in vitro studies to see what happens, what could possibly underlie this um, gain of trisomy 20. Our pet theories are, of course, initially we wanted to know whether it was H. pylori, but that given that it's so prevalent and quite has a significant role in the origin of stomach cancer. Um, but the data that we had on H. pylori status in our donors was patchy at best. Plus, of course, we're working with the fact that these trisomies happened a long time ago. So I think some people are following up, especially SY, who's the gastric cancer expert from Hong Kong that's central to this study. I think she's following it up um, to see what she finds in her organoid models as well. Yeah, thank you.